In this section, we're going to talk about the sensory receptors, specifically the cutaneous receptors. So, so sensory receptors, basically, their job is to encode data into neural impulses so our brain can understand what's going on around us. So the sensory receptors, they are located at the distal ends of peripheral neurons, and each type of receptor is specialized. It responds to only a specific type of stimulus of adequate intensity under normal conditions. So if you were come in from the cold, your hands are really cold, and you run your hands under the water and it feels super warm, no matter what the temperature of the water is. So those are sort of not normal conditions. Your sensory neurons are not responding the same as they would once you come up to room temperature or body temperature. But the main thing is each receptor is specialized and responds only to a specific type of stimulus. So there are three categories and then one sort of subcategory that we'll talk about. The three categories are mechanoreceptors, chemoreceptors, and thermoreceptors. So mechanoreceptors, they respond to the mechanical deformation of the receptor by touch, pressure, stretch, or vibration. Chemoreceptors respond to substances released by cells, including damaged cells after injury or infection. So remember in patho we talked about during the inflammation process how the cells release histamines. Well, your chemoreceptors respond to those histamines. Thermoreceptors, they encode and transmit information regarding heat or cold. So the subset of receptors you can think of are nociceptors. And nociceptors, they can encode any of these types of information, except it has to be to the intensity that it might cause tissue damage. So nociceptors can be thought of as damage receptors, and mechanoreceptors respond to mechanical deformation of the receptor. Chemoreceptors respond to chemicals. And thermoreceptors respond to heat or cold. So besides nociceptors, all the other ones, their names tell you what they respond to, which is pretty handy for remembering those things. So that's what nociceptors really, they are responsive to stimuli. The mechanoreceptors and chemoreceptors and thermoreceptors all have particularly sensory endings that we'll talk about a little bit that allow them to encode certain information. The nociceptors tend to be free nerve endings, so they are looking for tissue damage. That's kind of their specialty area. So they might detect ischemia or stretch that's to the point where it's going to cause damage, or they might detect some of these other areas. So the somatosensory first-order neurons, so we talked about how the first-order neurons go from the periphery to the spinal cord, and then the second from the spinal cord to the thalamus, and the third order from the thalamus to the cerebral cortex. So the somatosensory first-order neurons have two axons. So that's that pseudo-unipolar thing that we talked about in Chapter 5. The distal axon conducts messages from the receptor to the cell body, to the soma. And the cell bodies are located in the dorsal root ganglion. We'll talk more about that. But the proximal end of the somatosensory first-order neurons, the proximal axon, projects from the cell body into the spinal cord or brainstem. So the peripheral somatosensory afferents, which are going towards the brain, are classified according to axon diameter. So this chart from the book, it shows it from 1A's to C's, from largest to smallest, if you want to think of it that way. The large diameter axons transmit information faster than smaller diameter axons. So that's really the functional significance. The bigger, the faster. The smaller, the slower. So also the degree of myelination. The bigger axons, the 1A's and the 1B's, are more myelinated, and the C fibers are less myelinated. So sometimes you'll see them called unmyelinated, and we really don't have very many axons in our body that are completely unmyelinated, but you can think of it as less myelinated. So it's the slow route. It's taking longer because it's a smaller axon and there's less myelination. So we talked about there being two different kinds of somatosensory innervation, cutaneous and musculoskeletal. So we're going to talk about cutaneous, and then the next section we'll get into musculoskeletal. But in cutaneous innervation, which is the innervation of the skin, the receptive fields tend to be smaller distally and larger proximally. We saw an illustration of this in Tests and Measures lab when we were doing 2-point discrimination. So distal regions of the body have a greater density of receptors than proximal areas. So the combination of small receptive fields and greater density of the receptors distally enables us to distinguish between two closely applied stimuli on a fingertip. However, the same stimuli will be distinguished just as one instead of two on the trunk because it has larger receptive fields. So in the diagram, you can see that caliper A, it has both legs in the same receptive field, and that's going to be perceived as one single point. Caliper B has a leg in two different receptive fields, and that's going to be perceived as two separate points. So if the receptive fields are smaller and more dense, then you're going to have better discrimination. So the tips of our fingers obviously have better sensory discrimination than our trunk, because that allows us to go out and explore the world with our hands, which is kind of cool. So the sensations from the skin include touch, nociception, and temperature. So touch information is categorized as light touch or coarse touch, or fine touch or coarse touch. So fine or light touch includes a variety of receptors. So this is diagram 10.4 from page 187 in the book. And you can see the light touch receptors like the Merkel's discs and the Meissner corpuscles. They're close to the top of the skin, the outer surface of the skin. They are going to respond to stimulation from light touch. The sub-sensations that include vibration and skin stretch and skin pressure, those are all those deeper ones, the Pacinian corpuscles and the Ruffini endings. You can see how in order to stimulate that receptor, you would have to have some pressure or some vibration. The Ruffini endings, you can think of as sort of little crumpled up things, and they respond to stretch when they're uncrumpled. So they respond to a specific stimulus based on their physical attributes. The free nerve endings are specialized to respond to coarse touch and nociception. And of course we know that nociception is responding to stimuli that can damage or threaten tissue. So coarse touch provides information perceived as like tickle and itch is a coarse touch thing. So you can think of like tickle and itch could sort of be an irritating sensation. Fine touch can be a nice sensation. So if you kind of want to separate it in your mind that way, coarse touch is those free nerve endings, and that's like tickle and itch and ooh, what's that thing crawling on me? And fine touch is more like, wow, that feels nice. So that's just perception, but I'm trying to come up a way for you to think of it in your mind. So cutaneous receptors aren't proprioceptors, but the information from cutaneous receptors also contributes to our sense of joint position and movements. The contribution of cutaneous receptors is primary kinesthetic, responding to stretching or increasing pressure on the skin. So we talked a little bit about dermatomes last summer in your Tests and Measures lab, and distinguishing between a peripheral nerve lesion and a spinal nerve root lesion is important to target the right structures with our interventions in physical therapy. So the dermatome is the area of skin innervated by axons from a single spinal dorsal root. Dermatomes are usually used to diagnose lesions affecting a single nerve root and to determine the sensory level affected by a spinal cord injury. So you see the dermatome diagrams. There's one right here. It's pretty small, so you can look at the one in the book, which is bigger. It's not going to be exact, but it's pretty close to what you're going to see. For the limbs, there are two distinct distributions of sensory innervation, peripheral nerve and dermatome. If a lesion involves a peripheral nerve, the sensory impairment will be in the distribution of that peripheral nerve. However, if the lesion involves a spinal nerve root, the sensory impairment will be in the distribution of the dermatome instead of the peripheral nerve distribution. So the difference between peripheral versus dermatome innervation is used to determine the source of the lesion, basically. So the distribution of sensory impairment allows the PT or the doctor to accurately identify the location of a lesion. And the dermatome distribution indicates spinal nerve root involvement, whereas the peripheral nerve distribution indicate peripheral nerve involvement.